Yeah, so today we are going to discuss the second part of cell biology, that is cell division. Why cell needs to divide is the most important question, and for that there are so many reasons for cell to divide. Okay, so what actually the cell division is? Cell division is actually the process where cell grows in number, grows in their number. Okay, so cell division, as I said, is happens for various reasons. Cell division has so many reasons to occur. The reasons may be include, okay, one maybe for repair. What is repair and all these things we'll discuss in a short while. Okay, or else for growth, or it can also be because for the sake of development. Okay, so there are various reasons for cell division. What are they? Either for repair or for growth or for development. Okay, so in our daily life, so many cells in a human body or any other organism, so many cells will be dying and they will be replacing with the new cells. So the new cells have to be replaced. There must be a mechanism. There must be a mechanism for the new cells to regrow. So that mechanism is nothing but cell division. Okay, so sometimes you may come up with some injury, and that injury has to heal up, and the healing also occurs because of the cell division. If cell division is not there, there won't be any wound healing. So the cells which are hampered because of the injury or some other accident, so they have to be repaired and replaced. Those which are not which are not useful for the body have to be replaced. Okay, so and at the same time the cells have to grow. Okay, from as you know, our we see the first stage of the any organism is starts with only single cell. Okay, single cell starts the life process, that is zygote. Okay, only zygote starts life cycle, and after that there are so many blastula stage, gastrula stage, then tadpole stage. All these stages will be uh, part of our life cycle. So for all these stages, for the growth of the human being, or else for growth of any other organism, the only thing which is needed is cell division. Without division of cells, there won't be any wound healing, there won't be any development of the organism, or else there won't be any growth in organism. So now today we are going to discuss how the cell division occurs, and uh, what are the different steps involving in cell division. Okay, broadly cell division means there are two main important things. There are only two important things. Okay, two important things in cell division. So the two important things in cell division, they are, what are they? They are, first one is karyokinesis and cytokinesis. What is karyokinesis? Karyokinesis is division of, caryon means, as I said in the last class, caryon means nucleus. Okay, Kinesis means division. Okay, division of nucleus is known as karyokinesis, and at the same time division of cell is known as cytokinesis. Cyto means cell, kinesis means division. So division of cell is known as cytokinesis and division of nucleus is known as karyokinesis. These are the two important steps in cell division. Okay, so what are the different steps in karyokinesis and when the cytokinesis occurs? These are the two important things. Which one will occur first and which one will occur later, at the later stage, are the things which we have to decide, we have to discuss now. Okay, so first karyokinesis occurs. That means the cell inside the nucleus will divide into two, and then after the nucleus divides, then the cell will be dividing into its respective parts. Okay, so cell division again, based on the tissue, based on the cell where it occurs, is again of two types. Cell division is again of two types. What are the steps in cell division? Karyokinesis and cytokinesis. And the cell division is of two types. One is mitosis, and second one is meiosis. Okay, today we are going to discuss only about mitosis, and will be discussing meiosis separately in a in our next video, as the meiosis is a lengthy topic. So we'll be discussing the general topics of cell division and also mitosis in this video, and next video we'll be discussing about the meiosis. Okay, so what is mitosis? What is meiosis? Mitosis is also known as equatorial division. What? Equatorial division. And meiosis is reduction division. Reduction division. Mitosis is equatorial division and meiosis is known as reduction division. Why they are called so? Okay, so it is called equatorial division because when the cell starts to divide, if suppose the cell starts with 4 chromosomes, and at the end of the cell division there are 4 chromosomes with two daughter nuclei. Okay, so the number of chromosomes will not be changing in the parent cell or in the daughter nuclei. That is why it is known as equatorial division. And meiosis is known as reduction division. Why? Because if the cell starts with 4 chromosomes, but it ends up to have only 2 chromosomes in daughter cells. That means parent cell will be having 4 chromosomes and daughter cell will have only 2 chromosomes. That is, half of the parental chromosomes will be present in the daughter chromosome. That means there has been a reduction in the number of chromosomes from the parent cell to the daughter cell. That is why it is known as reduction division. Okay, I hope you understand why they are named as equatorial division and reduction division. Okay, so as I said, we'll be discussing only mitosis part today, and meiosis part will be discussing in our next chapter, next topic. Okay, so what is mitosis and what are the different stages of mitosis we'll be discussing. As I told you earlier, mitosis is also known as equatorial division, and one most important thing regarding mitosis and meiosis is mitosis occurs only in vegetative cells or somatic cells. Vegetative or somatic cells. Vegetative cells or somatic cells. So those cells which are not, which are only vegetative are somatic. I hope you know what somatic and what vegetative is. Okay, so somatic cells are those cells which does not involve in any reproduction, and they are the cells which doesn't which involve only in regular activities. Okay, the skin cell, the digestive system cells, the lung cells, the heart cells, the epidermal cells, epithelial cells, all these cells come under somatic cells. Okay, mitosis occurs only in vegetative cells or somatic cells. Okay, as I already told you, it is equatorial division. Why it is known as equatorial division? Because the number of chromosomes doesn't change in parent cells. Number of chromosomes doesn't change in mother cell and also in daughter cells. That is why it is known as equatorial division, and it is very common division, very common and regular division. Whatever happens to our body, that uh, during during digestion, so many cells will be dying, and during injury, a small injury or any other thing occurs in normal cells, those cells will be healed up and replaced by only this vegetative, all these vegetative cells will be replaced only by this mitosis division. Okay, so but meiosis is reduction division, and this occurs only in germinal cells. What cells? Germinal cells. Okay, what are these germinal cells and how meiosis occurs, what are the different steps, we'll be discussing our next class, next video. Okay, so what are the different steps in mitosis? Okay, mitosis has four phases. One thing you should remember, these are all the man-made phases. Okay, only for our own distinguishness, only for our comfort, we have categorized these steps into different phases. But actually in real life, what happens, these are all a regular course of events. There won't be any time gap between these events. Okay, so for the cell division, you people have also have to understand what is cell cycle, and we'll be discussing cell cycle in forthcoming videos. So cell cycle is also a major important concept. Only when you understand what cell cycle is, then only you people can understand what cell division is. Okay, for this reason you have to go through cell cycle, and this cell cycle will be discussed in forthcoming videos. Okay, well, for the time being, you just remember that cell cycle has different phases. Those phases are G1, S, G2, and mitosis or meiosis, and G0. Okay, gap 1, synthesis, G2, and M phase. Okay, cell uh, cell DNA gets doubled in synthetic phase. DNA gets doubled in synthetic phase, and gap 1 and gap 2 phases are those phases where the gap will be for checking whether the DNA has been completely doubled or not, and are there any mutations or something. Okay, and M phase, M phase denotes either mitosis or meiosis. Okay, mitosis or meiosis based upon whether it is a vegetative cell or a germinal cell, and also whether it is a regular division, equatorial division, or it is a zygote which is being divided. Okay, so G0 is a phase where, which is also known as quiescent phase, and all these details will be discussed in cell cycle chapter. Okay, so you just remember G1 as G2, G1 phase, gap 1 phase, S phase synthetic phase, G2 phase, gap 2 phase, M phase mitosis or meiosis phase, G2 G0 phase is quiescent phase where cell will be in its resting stage. Okay, so what are those four phases of mitosis? Let's come to mitosis. What are the four phases of mitosis? Prophase, metaphase, anaphase, and telophase. Prophase, metaphase, anaphase, and telophase. Prophase and metaphase also will have a mm, intermediary phase, which is also known as prometaphase. Sometimes it is also given as prophase 1, prophase 2, early prophase, late prophase. So in every textbook will be giving its own interpretations. Okay, let's say that a phase which is in between prophase and metaphase is known as prometaphase. Okay, so prometaphase, either you can also say it as late prophase. Okay, prophase, metaphase, anaphase and telophase are the four important phases of cell cycle. Okay, so as I told you in cell cycle description, this chromosome doubling occurs in S phase. S phase is also known as interphase or synthetic phase. Interphase or synthetic phase. Okay, so now all these duplicate chromosomes, when the chromosome gets doubled, all these duplicate chromosomes will be held together in pairs. Okay, held in pairs. Okay, suppose this is a chromosome, it gets doubled and they are all held in pairs. Okay, so these pairs are known as sister chromatids. These pairs are known as sister chromatids. Okay, so now let's discuss all the phases in detail. All this, what happens to this sister chromatids. First, okay, in G2 phase, once the synthesis gets completed, and when the cell enters from G2 phase to M phase, the cell will be something like this. Okay, cell will be something like this. Spindle fiber formation gets started. Okay, spindle fiber formation gets started, and also the nucleus. Inside the nucleus there will be a nucleolus and chromosomal condensation. Chromosomal condensation will be starting in the nucleus. Okay, so this will be the cell in G2 phase. In G2 phase, the cell's condition will be something like this. Okay, now after this, what happens is, in the first phase of our cell cycle is prophase. In prophase, how the cell will be looking like. Okay, just let's see with a diagram. The cell, in the cell, the chromosomal condensation completely starts and nucleolus gets disappeared. Chromosomal condensation, that means chromosomes will be will become thicker in their structure, and the nucleolus will be disappearing from the site of nucleus. And at the same time, the mitotic spindle fiber, whichever has been the initiation of mitotic spindle fiber just have been started in G2 phase, will be getting extended in this phase. Okay, getting extended, and also at the same time the nuclear pore will start disappearing a little bit. This is early prophase. In the late prophase, what happens is, completely this nuclear envelope whichever is present, this nuclear envelope will completely be degraded, and the chromosomes will be out of this nucleus into the cytoplasm, and all the cell organelles in the cell will be disappeared in this phase. And this what we said, this spindle fiber, this mitotic spindle will be taking over the chromosomes. Okay, all the chromosomes in the late prophase, the cell will be looking like something like this. Mitotic spindle will be taking over in a cell at the poles. Okay, at the poles, mitotic spindle will be starting to take over. Okay, in this way, in this manner, Okay, and also at some or other place, as I told you, at the kinetochore, the chromosomes will be having a centromere, and the centromere is a region where the sister chromatids will be attached to each other. At this site, at the kinetochore, the chromosome, the spindle fiber attachment takes place, starts. Okay, spindle fiber gets attached to the kinetochore at different places. At one or other place they will be attached, and with those which doesn't attach to any those spindle fiber which doesn't attach to any chromosome will be attached to each other, and they will be stabilizing the cell in this phase. Okay, this is prophase late prophase. Okay, after this, in late prophase, or as I told you, it is metaphase. Prometaphase, condensation will completely be finished. Condensation of chromosomes will be completely finished, and mm, new spindle fibers gets attached to the chromosomes at one or other place, and nuclear envelope is completely broken down. It doesn't appear anywhere in the scene. Okay, then the next phase is metaphase. What happens in metaphase? How you can understand what is metaphase? In metaphase, the cell will be, okay, attachment to the spindle fibers. Something cell will be looking like this, something like this. Attachment to the spindle fiber with the chromosomes will be almost finished, and all the chromosomes will be lying in a single plane. In a single plane. In a single plane, all the chromosomes will be attached. Okay, and this is known as metaphase plate. Metaphase plate formation is done over here. All the chromosomes will be lying in a single plane in a horizontal way. Okay, so all the chromosomes get attached to spindle fiber at kinetochore, as I told you, kinetochore. And then this at this position, the cell will be checking out whether all the spindle fibers got attached to the chromosomes and all the chromosomes are attached to the spindle fiber or not. Where if there are any leftover chromosomes are present in the cell, this is known as spindle checkpoint. Okay, once the if the cell detects that there is some discrepancy in spindle checkpoint, the cell halts over there, and cell gives the time to the mechanism so that the metaphase plate formation gets completed and metaphase is over. Okay, until or unless all the chromosomes attached to the kineto by kinetochore to the spindle fibers, there won't be any further advancement in the cell. So this is known as spindle checkpoint. Okay, next phase is anaphase. In anaphase, here the chromosome gets pulled by the spindle fibers. The chromosomes gets pulled by the spindle fibers to the opposite directions, and the sister chromatids gets detached to each other from each other. The chromosomes will be detached, and the chromosomes will be pulled towards the ends of the cell, two ends of the cell. At the same time, towards the horizontal plane, two notch like structures will be appearing. Okay, these are for cell division, that is cytokinesis. notch-like structures will be helpful for cytokinesis. Okay, now these notch-like structures further gets furrowed and gets furrowed completely, and spindle fiber gets broken down. Spindle fiber gets broken down. Spindle fiber gets broken down and the notch-like structure gets deepened, and nuclear envelope starts reappearing. Nuclear envelope reappears. Nuclear envelope starts reappearing. Okay, this is anaphase and this is telophase. Okay, finally, after this telophase, cytokinesis is the result of the cell mitosis. What happens? Two different cells will be formed, okay, with the same number of with the same number of chromosomes, and the nucleolus reappears and the condensed chromosome gets decondensed again. And finally two daughter cells will be formed from one large cell. Okay, these are the daughter cells. They also will be having similar number of chromosomes as in case of parental chromosome. Okay, so this is the complete picture of mitosis. Mitosis is also known as equatorial division. Why equatorial division? Because the number of chromosomes present in the parent cell and number of chromosomes in the final daughter cells is equal. There won't be any change. And after this, all the cell organelles starts reappearing and the cell starts functioning as usual. Okay, after this the cell finally detects whether to get into the G2 phase, or either cell gets into G2 phase and then to S phase, or else the cell will decide, the mechanism of cell will decide whether the cell to get into quiescent phase, which is also known as G0 phase. Either cell will go to G0 phase or cell will go to G2 phase. Okay, cell will either go to G0 phase or will go to G2 phase. If the cell gets into G2 phase, then cell will get into the synthetic phase and then G1 phase and finally to M phase again. This M phase again will be again a mitosis. Okay, this is a sequence of sequential events which are present in mitosis. Present which will be present in mitosis. This can be the general experiment we conduct for the study of mitosis is Allium cepa roots of Allium cepa. Okay, in Allium cepa roots there will be very good structures, different chromosomes will be appearing very nicely. Okay, so this is regarding mitosis and cell division. In the next video we will be discussing about meiosis. If you like this video, please subscribe to my channel, and also if you like this video, please like, share, and if you have any doubts, please comment in the below comment section. I'll be happy to addressing you all. Thank you very much.